Hi, we are from B1 M3P6. We are going to talk about recombinant DNA technology.
Hi everyone, I'm Manissa. What is recombinant DNA technology? The study of techniques and procedures in gene manipulation whereby researchers join together DNA from different organisms in the laboratory to produce new genetic combinations that are valuable to science, medicine and agriculture industry. Where can we apply recombinant DNA technology? One of the applications is medicine, which produce insulin. Human insulin was the first human protein manufactured by gene cloning for human use. Human insulin produced by recombinant DNA technology was the first commercial healthcare product derived from this technology. There are 5 tools used in recombinant DNA technology. The first one is target DNA. Second one is restriction enzyme. Third one is DNA cloning vector. Fourth one, host cell. Lastly, modifying enzyme. The first tool is target DNA. Target DNA contains the gene of interest. Target DNA will be inserted into a vector to form a recombinant DNA. So Afiq, what is the next tool?
So the next tool is restriction enzyme. What is restriction enzyme? Restriction enzyme is which cleave or cuts DNA molecules at restriction site produces short restriction fragments which produce sticky end or blunt end.
E. coli recognizes the base sequence and makes staggered cuts within the sequence and produce sticky ends. Allow for DNA ligase to ligate and producing recombinant DNA.
Wow, such a long explanation, Afiq. Next is modifying enzyme. There are DNA ligase and tag polymerase. Next,
the third tool is DNA cloning vector. DNA molecule which can insert it with gene or fragment into a cell and replacing that cells. There are 3 characteristics as shown. We take plasmid as an example. Why? Because the DNA molecule can carry foreign DNA into a host cell and replicate there. The capacity to carry insertion size is 1 to 10 kb.
Fadhilah here, as the last tool is host cell. Host cell is a cell which accepts the foreign DNA and allows the structure to maintain recombinant DNA. There are 3 characteristics as shown. As an example of host cell is bacteria for E. coli. This is because DNA can be easily isolated from or introduced into a bacterium.
Moving on to gene cloning. So Manissa, what is gene cloning actually? Gene cloning is a process in which a gene of interest is located and copied out of DNA extracted from an organism.
there are 5 steps in gene cloning.
The first one is the isolation of gene.
Second, cleave or cut.
The third one is insertion.
Fourth, transformation and amplification.
And the last one is screening.
The first one is isolation of gene. Plasmid from bacteria cell is isolated. Gene of interest is isolated from DNA of donor cell.
Next is cleave or cut. Donor DNA and plasmid bacterial is cleaved using compatible restriction enzyme. Make a single cut within the exact gene site of cleaving, producing complementary sticky end.
Next, insertion. DNA fragment is inserted into the plasmid, joined together by DNA ligase, forming recombinant DNA.
The fourth step is transformation and amplification. The recombinant DNA then inserted into the bacteria through transformation process. As the bacteria reproduce, the recombinant DNA is also replicated or we call as clone.
The last step is screening. Screening is to identify bacteria clone containing recombinant DNA. Blue-white screening is done. How? Place the bacteria on alga containing antibiotic, ampicillin, and sugar, or known as Asst. Incubate until colonies grow.
Blue-white screening technique. Place the bacteria in agar containing antibiotic, ampicillin, and Asst. It will produce 2 antibiotic ampicillin. The nut-resistant antibiotic ampicillin will not make the bacteria grow.
But resisting the antibiotic ampicillin will make the bacteria reproduce and form colonies.
It will form two colonies. Colonies without recombinant plasmid will be blue. It contains intact lactate, which can produce big galactosidase and can hydrolyze as GAR.
While colonies with recombinant plasmid will be white, do not contain intact lactate, cannot produce big galactosidase and cannot hydrolyze as GAR.
We hope all of you could understand what we explained. Let's take a look at the summary of this process.